Now we are going to start the structure of bacteria or living microorganisms, and we must say what are the characteristics of life. How can you distinguish between one organism that it is a living organism or it is just a particle? The first thing, it must be able to grow and develop on its own. The second thing, reproduction and heredity. The genome, composed of DNA packed in chromosome, produced offspring sexually or asexually. So it must be able or have the ability to give an offspring or divide either by meiosis, mitosis, or by budding, anything, but it must have the ability to multiply and grow in number. And this must be done by the genome of the microorganism. The third thing, it must be able to make metabolism, chemical or physical life process must be done through metabolism. Motility or irritability, it must be able to respond to stimuli, either go toward something or run away from something, move in the environment is a sign of life. Cell support, protection, storage mechanism, cell wall, vacuoles, granules, and inclusions. All these are forms of life. Transport of nutrients and wastes, transport of nutrients inside the cell, and transport of waste products outside the cell. This is a form of life. So any living organism must include these forms of life. We can see the structure of the prokaryotic cell and the eukaryotic cell. As we said, we are concerned with the prokaryotic cell, and we are going to see every single detail of the structure of the prokaryotic cell. But we are interested to know what are the difference between the two cells. As you can see, there is a cell wall in the prokaryotic cell. It's not there in the eukaryotic cell. The eukaryotic cell have only cell membrane from the outside, and this is a very good thing because antibiotics that can act on the cell wall, they act only on bacteria and they don't have any action on the eukaryotic cell. This is the idea for many antibiotics like the penicillin, the cephalosporins, all of these work on the cell wall. That's why they are effective on the prokaryotic cell and on the bacteria, but not on the human cell. Other thing, you can see there is a difference in the chromosome. It's only one single chromosome. It's a big circular one, double-stranded DNA in the case of the prokaryotes. In the case of eukaryote, you can find that the chromosome is present inside the nucleus. Here it's not. There is no nucleus, only chromosome in the cytoplasm in an area called nucleoid. In the eukaryotic cell, the genome or the genes, it is inside the nucleus. There is something here called endoplasmic reticulum. There is mitochondria. There are the Golgi bodies. All of these are present in the cytoplasm of the eukaryotic cell, but not in the cytoplasm of the prokaryotic cell. Ribosomes can be found in both types of cells. There are flagella found in the prokaryotes, not in the eukaryotes, and so on. Prokaryotes, they do not have membrane surrounding their DNA. They don't have nucleus. They have single circular chromosome found in the cytoplasmic region called nucleoid. So the region in the cytoplasm that have the chromosome is called nucleoid. The prokaryotic cell lack various internal structure, no endoplasmic reticulum, no Golgi bodies, nothing. They divide by binary fission as we saw before, where this involves fewer structure and process than eukaryotic cell division. They have a simple structure. They are smaller in size, in diameter than the eukaryotic cell. They have small ribosomes, and it's called 70S in case of the prokaryotic cell, sorry. In the eukaryotic cell, it's called 80S. And as we know, the prokaryotes are comprised of bacteria and archaea. The eukaryotes have DNA found in the cell-bound nucleus, cell membrane-bound nucleus, and the DNA is found in multiple chromosomes, not single, one as the prokaryotes. DNA is associated with chromosomal proteins called histones, and these histones they are rarely found in the prokaryotes. They have internal membrane-bound organelles like the mitochondria and plasmic reticulum, Golgi bodies, and so on. They are larger in size, can reach 100 micrometer in diameter, and it's more complex in structure. The ribosome are larger, ATS. The cell division is by mitosis, and there are many examples for eukaryotic cell like the algae, protozoa, fungi, animals, and plants. The bacterial cell structure, it is composed of three main parts, as we can see. The external part and includes the appendages, flagella, villi, and fimbriae, glycocalyx, which includes the capsule and the slime layer, the middle part, which includes the cell wall and the cell membrane, and as we said, there is no cell wall in the eukaryotic cells, and the internal parts, which includes the cytoplasmic matrix, which includes all the things inside, like the ribosome, the inclusions, and the chromosome, and the endospores. As you can see, this is the bacterial cell structure. If you go from the inside to the outside, you can find the cytoplasmic matrix, including the inclusion bodies, the ribosomes, the nucleoli. Then you can find the cytoplasmic membrane, and outside the cytoplasmic membrane there is the cell wall. So after that there is the cell appendages and the glycocalyx part. If you look at this table, you can find the function of every single part of the cell. Like the cell wall, its main function is to give shape and for protecting the cell against the environment around it, and so on. Glycocalyx is the first part in the external structure. It's made mainly of a sugar coat, in the general term used for substance that surround the cell. It's the very outer layer of that cell. You can find appendages after that. It's two major groups of appendages. Either the appendages are used for the motility, like the flagella and axial filaments, or they are used for the attachment, like the fimbriae and villi. In the glycocalyx, the bacterial glycocalyx is viscous and sticky gelatinous polymer that is external to the cell wall and composed of polysaccharide, polypeptide, or both. So the main constituents of this glycocalyx is the polysaccharide and polypeptide. The types of this glycocalyx, it can be either slime layer or capsule. You can see here there is a difference between the shape of the slime layer and the capsule. The slime layer, it is loosely organized, okay, and is loosely attached to the bacteria, unlike the capsule, which is highly organized and tightly attached to the bacteria. So if a bacteria is surrounded by slime layer, you can find the outer layer, it is not entire, okay? However, if it is surrounded by a capsule, you can find it completely entire and smooth around the microbe. The main function of the glycocalyx is to protect the cell from dehydration and losing the nutrients, inhibit killing by white blood cells or by phagocytosis, so it increases its pathogenicity. It provides also attachment and the formation of biofilm. The bacteria can form biofilm if they have this slimy layer or this glycocalyx. They become attached easily to any surface, and they can form the biofilm very easily. The slime layer is most of the time glycoprotein and loosely attached to the cell surface. These slime layers cause bacteria to adhere to solid surface and help prevent the cell, as we said, from the drying and losing the nutrients. They are sticky layers, as we said, and they allow biofilm formation. For example, Streptococcus, they form slime layer. They are gram-positive microorganism. One of the species is the Streptococcus mutans. It allows it to accumulate on the tooth enamel and is one of the causes of the cavities. بتسمح السلايم لاير دي بتسمح للميكروب ان هو يجي ويلزق على الانامل، طبقة الانامل اللي في الاسنان، بالتالي بيمنع دخول الاير للاماكن دي وبيسمح بدخول other بكتيريا، معظم الوقت بقى anaerobic بكتيريا اللي بتعمل الكافيتيز وبتعمل التسوس. Other bacteria in the mouth become trapped in the slime layer and form biofilm and eventually build a plaque. طبقة البلاك اللي احنا بنروح لدكتور السنان عشان يشيلها لنا، دي بتبقى سببها بكتيريا معينة زي ما احنا فهمنا دلوقتي، بتعمل slime layer وبتعمل بايوفيلم فبتعمل طبقة البلاك. Staphylococcus, the slime layer of gram-positive Staphylococcus allows it to thrive in the salty hypertonic environment of the skin and protect them from desiccation and disinfection. As we can see, these bacteria, they have the ability to form slime layer and they get attached to the skin, and it becomes resistant to the use of disinfectants because of this slime layer that is formed by the microorganism. The other type of the glycocalyx is the capsules. Capsules are important in contributing virulence to the bacteria. That means it increases its ability to cause diseases. Capsule can protect a bacterial cell from destruction by white blood cells. It can protect it from phagocytosis. It makes the bacterial surface component more slippery, helping the bacterium to escape engulfment by the phagocytic cells. So it can escape easily from the phagocyte due to its slippery outer surface. You can see these capsules in the lab. I think you will see them as spots in the lab. It's very easy to stain them and see them under the oil immersion lens. Bacillus anthrax produce a capsule of D-glutamic acid. Only encapsulated B anthrax causes anthrax. So, as we can see here, the pathogenicity of this Bacillus anthrax is due to formation of the capsule. The uncapsulated organism, they don't cause the disease. Only the capsulated Bacillus anthrax cause the anthrax disease. Another example is the presence of the polysaccharide capsule in the Streptococcus pneumonia. It is the most important factor in the ability to cause pneumonia. Pneumonia, it's one of the respiratory tract diseases. and they found that only the entire or the soft or species of the Streptococcus pneumonia, it is the only candidate that causes the pneumonia disease. Otherwise, the other species which do not have capsule, it doesn't cause pneumonia. The polysaccharide capsule of Klebsiella also prevents phagocytosis and allows the bacterium to adhere to and colonize to the respiratory tract. Also, the Klebsiella, it causes the pneumonia disease, and they found that the capsulated species only cause that disease.
Biofilms can cause a wide range of problems in building water systems, including cooling towers, decorative fountains, and domestic water. Biofilms are a complex community of problem-causing microorganisms encapsulated in a protective coating, sometimes called slime. Biofilms form when free-floating bacteria in the bulk system water attach to surfaces such as pipe walls, shower heads, chillers, and basin walls. Attached bacteria produce an extracellular polymeric substance or slime that holds the growing biofilm together. Biofilms are 90% water and very insulating when present on heat transfer surfaces. As biofilm matures, a complex community of microorganisms develop protected from biocide additions, including Legionella bacteria, and cause fouling. The number of bacteria in the bulk water do not correspond with the amount of biofilm. Microbiologically induced corrosion can occur underneath biofilms. In an advanced biofilm, higher life forms, such as amoeba and protozoa, ingest bacteria in the bulk water and biofilm. Some bacteria, such as Legionella, can multiply inside the higher life forms to the point of bursting the host organism. This can suddenly release high levels of Legionella into the bulk water. Biofilms can cause a variety of problems in water systems, including corrosion, fouling, reduced heat transfer, and contribute to Legionella growth. Biofilms can cause a wide range of
After we saw the previous video, we can now describe what are biofilms. In nature, microorganisms seldom live in the isolated single species colonies. The isolated single species colonies is called planktonic. So most of the time you don't find these planktonic cells in nature as you find them in the laboratory. They more typically live in communities called biofilms. What is a biofilm? It is a complex aggregation of microorganisms growing on a solid surface in which cells are stuck to each other or to a surface. The glycocalyx is a very important component of this biofilm. A glycocalyx that helps cell in a biofilm attach to their target environment and to each other is called extracellular polymeric substance, or EPS. So what is EPS? It is a type of glycocalyx. Its main function is to stick cells together or to a certain surface. So in order to accomplish making this important biofilm thing, which is very important to debug. Mostly biofilms in nature, you can find them wherever there is a moisture. For example, Serratia marcescens, it formed biofilm on the urinary catheter or in contact lens wearers. The urinary catheter or elastara, you can find this organism attached to the catheter itself or to contact lens wearers. As you will see the picture later on, it easily attached also to the contact lens. These soft contact lenses have the composition of these lenses is mostly water. That's why it is easily attached to it. Streptococcus mutans, as we said, it is very important cause for the dental caries and the dental plaque. It attaches itself to the surface of these by the glycocalyx. And it forms this Streptococcus mutans, it forms this biofilm. That's why it forms this plaque and these dental caries easily. The CDC, or the Center for Disease Control, it estimates that over 67% of nosocomial or hospital-acquired infections are caused by this biofilm. Maybe you don't know what's a nosocomial infection, what's the right definition for it, and we'll say it later on. So most of the hospital-acquired infections, or the infection that you acquire in the hospital and that you were a certain disease, it is a certain disease that you are not admitted into the hospital with this disease. You were admitted for something else, and you acquired this disease during your stay in the hospital. Most of the time it's very resistant type of microorganism to deal with, and they found that it's most of the time it is caused by biofilm forming organisms. They can also be a problem in pipes and tubing where their accumulation impaired circulation. That means that it plugs or it stops the flow of water inside the water pipes. It totally plugs these water pipes, and you can see it during the work of the plumber. He gets black stuff out of the water pipes during his work, and these things are the slimy things that you can see are due to the biofilm forming bacteria. Cell-to-cell chemical communication or quorum sensing allows bacteria to coordinate their activity and group together into communities that provide benefits. What does that mean? It means that what is the language between the bacteria and the different members of bacteria? It's something called the quorum sensing. They communicate with each other through certain chemicals or that are carried on by certain signals. Okay. After this communication of different members of the bacteria together, they can set an agreement to attach to this certain surface and form their biofilm in order to accomplish what they want to accomplish. Biofilms are an important factor in human health. For example, microbes in biofilms are probably thousands times more resistant to microbiocides. It's not easy to deal with diseases that are caused by biofilm. That means that if you take, for example, certain antibiotic course to deal with a regular disease, it needs more time, or it needs more powerful antibiotic to deal with that same organism if it's in biofilm. Most healthcare associated infections are probably related to biofilm in medical catheters. Biofilms form on almost all indwelling medical devices, including mechanical heart valves. It's a common problem that all the prosthetic or the things that you embed into the human body, like the medical catheters or the valves that it's in the heart, or in the joints, the artificial joints. This organism, the biofilm forming organism, they can indwell into these devices, and some of the times you cannot take a regular antibiotic course and get rid of this biofilm forming organism. No, you need to remove this artificial knee or this artificial catheter, wherever it is, and go into surgery and put another one. So it's not easy to deal with it. Biofilms can include those formed by fungi such as Candida. It can be encountered in many disease conditions. Example, infections related to the use of contact lenses, dental caries, and infections by Pseudomonas bacteria. It's very popular that you find these organisms on the catheters and on the soft contact lenses and the dental caries, as we said. Here you can see the organism in biofilm on the dental plaque, and as we said, also on the soft contact lenses. Contamination of the lens cases by bacteria, fungi, and amoeba is common in 20 to 80% of the lens wearers, which is a very high percentage having a contaminated lens case. We can say, how does this happen? You know, for any contact lens wearer, she or he, they have to wash their case regularly with regular tap water. And the regular tap water, it has what's called amoeba, acanthamoeba, a certain type of amoeba. This, if the case is not dried well after washing it with the tap water, this acanthamoeba will attach very easily to your contact lens. And when it is attached to the contact lens and then you put it on the cornea, it goes to the cornea, and it can form an ulcer, which is very difficult to deal with. If you don't treat it in the right way, it can make a severe damage to the cornea, and you have to go into corneal transplantation and a very big surgery. So it's better to wash your case, then dry it thoroughly, very well, with a clean tissue paper. And when you are washing your soft contact lens, you have to rub it between your palm and your index finger, so anything attached to the lens, it can go off easily. Otherwise, if you leave it, you cannot. Many layers of this acanthamoeba will be over the lens, and it will be very hard to get rid of after that. As we said before, nosocomial infection are caused by bacteria in biofilm. So what's a nosocomial infection? It can also be called the hospital-acquired infection, or HIE. This is the abbreviation. Infections are considered to be nosocomial if they first appear 48 hours or more after hospital admission or within 30 days after discharge. This is the case when you call it's a nosocomial infection. It can be either exogenous or endogenous. What's an exogenous? Exogenous, it comes from another patient or staff member or from the environment in the hospital. The environmental source could be in an inanimate object like a bed or a bench or whatever, air, water, food, cross-infection from another patient or from a hospital staff member suffering from infections or asymptomatic carriers. This is all called exogenous or from external organisms. The endogenous or the opportunistic normal flora, it comes from patient's own flora. The patient's own flora, it may invade the patient tissue during some surgical operation or a certain wound or an instrument manipulation. The normal common cell of the skin, the respiratory tract, it can cause this nosocomial infection. The most common nosocomial infections are the urinary tract infection, the catheter associated infection, pneumonia, bloodstream, and most important are the patients on the ventilators in the intensive care units. Nosocomial infection are enhanced by four main factors. The crowded hospital condition, the new microorganism that may be recently identified, increased number of people from compromised with compromised immune system, and the increased bacterial resistance. Microorganisms that can be encountered in the nosocomial infection or the hospital-acquired infection are any pathogens. Those able to develop resistance to antibiotics and disinfectants are the major causes for this nosocomial infection. Common cell bacteria from the normal flora of healthy people significantly protect by preventing the pathogenic microorganism from colonization. This is the normal case. However, it may cause infection in compromised patients. The most common bacteria that can cause this nosocomial infection are the coagulase negative staph, Staphylococcus epidermidis, and Candida. Pathogenic bacteria, they cause infection regardless to the host status. The prevention of this nosocomial infection can take place by antibiotic therapy to the carrier staph or the source patient himself. Proper asepsis of inanimate object, it should be undertaken seriously in order to prevent this nosocomial infection. Proper regular hand washing, but I say here proper, not only regular, but also proper hand washing, and it has to be done by a certain way. Sterilization of reusable equipments like the ventilators and humidifiers in the intensive care units. Use of face masks and gloves by the health care givers must be adopted in order to prevent this nosocomial infection. This diagram can summarize the sources of the nosocomial infection or the hospital-acquired infection. As we said before in the biofilm formation, that it's very important for the individual cells of the bacteria to communicate together, and we said that this communication is called quorum sensing. So quorum sensing is the ability of bacteria to sense and respond to environmental stimuli such as pH, temperature, presence of nutrients, etc. it has been long recognized as essential for their continued survival. So these bacteria sense the presence of a nutrient somewhere, and they can go for it. They communicate with each other and go to that place where nutrients are. Bacteria communicate with one another using certain chemical signal molecules. The bacteria leak a chemical into their surrounding. These chemicals are called autoinducers, which is a small organic molecule or peptide or lipid in nature. It differs gram-negative and gram-positive in these autoinducers. So in gram-negative, they employ an acyl homoserine lactone or alkyl quinolones and fatty acid methyl esters. However, in gram-positive bacteria, they use peptides like the auto-inducing peptides. So, quorum sensing is a process of bacterial cell-to-cell communication involving the production of and detection of extracellular signaling molecules, the autoinducers, as we said. Quorum sensing is a phenomenon that allows both gram-positive and gram-negative bacteria to sense one another and to regulate a wide variety of physiological activities. These activities include symbiosis, virulence, motility, antibiotic production, and biofilm formation, as we said. So, to abbreviate, the quorum sensing, it's nothing but the conversation between the individual members of the community of bacteria in a certain population. Back again to the bacterial cell structure, we talked about the external part of the external part to the cell envelope. Talked about the appendages. We talked just about the glycocalyx, and we are now going to start to talk about the flagella. Flagella, they are the organelles responsible for movement. They are long filamentous appendages that propel bacteria, or they make bacteria move from one place to another. Not all prokaryotes have flagella. Bacteria that lack these flagella are referred to atrichous, or what means they are without projection. The flagella is composed of three main parts: the filament, the hook, and the basal body. The filament is a long outermost region containing the protylflagellin, arranged in several chains that intertwine and form a helix around a hollow core. The filament is capable of rotating 360 degrees. The filament is attached to a slightly wider hook. This is the structure of any flagella. What's the hook like? It consists of different proteins, and the basal body is firmly anchors the filament and hook to the cell wall and plasma membrane. So it's the part that fixes the flagella to the body of the cell. The basal body is composed of small central rod inserted into a series of rings. The number of rings differs according to the type of the bacteria. So the main function of the flagella is the motility. It enables a bacterium to move towards a favorable environment or away from an adverse one. The movement of a bacterium towards or away from a particular stimulus is called taxis. So it may be chemotaxis if the stimuli is chemical, or phototaxis if the stimuli is the light stimuli. Flagella attachment in bacteria differ from one type to another. In gram-negative bacteria, it contains two pairs of rings. The two pairs of rings is anchored to various portions of the cell wall, and the inner pair of rings is anchored to the plasma membrane. So they are actually 4 rings in case of the gram-negative. This is because it has an outer membrane and an inner plasma membrane. In gram-positive bacteria, it has only one pair of rings which attaches to the plasma membrane because there is no outer membrane. The movement of the prokaryotic semi-rigid helical flagellum results from rotation of its basal body and is similar to the movement of the shaft of an electric motor. If you imagine a motor and this motor, it moves the shaft of this motor, it moves 36 degrees around. It's the same thing with the flagella of the bacteria. Some species of bacteria endowed with many flagella, like the Proteus. For example, it can swarm or show rapid wave-like movement across a solid culture media. If you see the culture of the Proteus, you don't find it regular movement, you find it like swarming. Here is the diagram for the flagella. You can see here in the gram-negative bacteria, there is the filament and the hook and the basal body. As we said, the basal bodies, it has 2 pairs of rings in case of gram-negative. One pair fixes with the plasma membrane, the inner plasma membrane, and the other pair with the outer part of the outer membrane. In case of the gram-positive bacteria, it doesn't have an outer membrane in the cell wall. It only has an inner membrane, inner cytoplasmic membrane. That's why it has only one pair of rings. According to the arrangement of the bacterial flagella on the surface of the bacteria, bacteria can be divided into 4 main groups. The peritrichus, where you find the flagella arranged all over the cell wall of the bacteria. The monotrichus and polar, you can find only one flagellum at one of the poles. Lophotrichus, you can find a bundle of flagella on one of the poles of the bacteria, and it can be also called polar. And the amphitrichus, you can find 2 flagella on both sides of the bacteria. These are the main 4 types of the flagella. And you can also find, as we said, what's called atrichus, which means the bacteria without a flagellum. Here are the 5 types of bacteria according to the arrangement of the flagella on it. Let's see this video and see what the movement of the flagella looks like.
Although flagella may appear to undulate back and forth like an eel swimming through water, they actually rotate 360 degrees, clockwise or counterclockwise. The basal body spins, much like the drive shaft of an electric motor, producing this motion. When the flagellum rotates counterclockwise, the bacteria runs in a single direction. When the flagellum rotates clockwise, the bacterium tumbles and changes direction. Movement toward or away from a stimulus is called taxis. The stimulus can be light in the case of phototaxis or a chemical in the case of chemotaxis. In either case, the bacterium contains receptors which send signals to the flagella. In the case of positive taxis or motion toward the stimulus, tumbles become less frequent when the cell moves toward the stimulus and more frequent when the cell moves away from the stimulus.
So that's what the movement of the bacteria with the flagellum looks like. We must differentiate between the flagella movement, or the bacteria that move with the flagella, and the Brownian motion. We all know what the Brownian motion is, which is the bombardment of the particles of media with each other. So if we the next video will show us the real movement of some kinds of bacteria or certain species of bacteria in the real medium. So it's clear here the real movement when the cell moves from one point to another, but the single cells that you saw that it's moving in splits, this is due to the bombardment with the particle of the medium. The other types of appendages are the fimbriae and the pillae. Fimbriae and pillae, they are hair-like non-motile appendages in gram-negative bacteria, so they are present only in gram-negative bacteria, not in gram-positive bacteria, and they are shorter, straighter, and thinner than the flagella. They consist of proteins called pilline proteins, arranged helically around a central core. You can see the difference between the fimbriae and the pillae. Here the fimbriae, they are the very small tubules around the cells, while the pillae, they are shorter, thicker than the fimbriae. Of course, they have different functions. Fimbriae, the singular is fimbria, may be hundreds per cell and can occur at the poles of the bacterial cell or can be evenly distributed over the entire surface. Function, its main function is adhesion to one another or to the host and to the substance in the environment used to attach the bacteria to the target cell in infection or to surface where they form biofilm. As we said, the formation of the biofilm to be formed, the cell must adhere first to each other or to a certain surface. So these bacterial cells adhere by the help or by the aid of these fimbriae. It's very important for infection. It's very important for the biofilm formation. Fimbriae clinically, they can help bacteria adhere to epithelial surface in the body. For example, the fimbriae on the bacterium Neisseria gonorrhea that cause help the microbe colonize mucous membrane. Once colonization occurs, the bacteria can cause disease. So that means only the species from the Neisseria gonorrhea which has this fimbriae, they can cause the disease gonorrhea. If they don't adhere to the mucous membrane, they cannot cause the disease. Fimbriae of E. coli 0157 enable it to adhere to the lining of the small intestine where it causes a severe watery diarrhea. So the pathogenicity of this species, of this E. coli, is dependent on the presence of this fimbriae. When fimbriae are absent because of the genetic mutation, colonization cannot happen and no disease can take place, of course. Pili, and the singular is pilis, rigid tubular structure, longer than fimbriae but shorter than the flagella. So, as we saw in the picture, it's much thicker than the fimbriae and the flagella, but it is much shorter than the flagella. Bacteria typically only have 1 or 2 per cells. Function is involved in the motility and in the DNA transfer by conjugation. As you will see in the conjugation process, in the genetics part, you will see that certain parts of DNA can be transferred from one cell to another through formation of pili between them. These pili are like pipes that attach two cells together, where the DNA can be transferred from one cell to another. In one type of motility called the twitching motility, it can be noticed in the Pseudomonas aeruginosa and in the Neisseria gonorrhea. This motility can be caused also by the pili. Also, the gliding motility can be shown by the pili in some species of bacteria. Here are the pili. As you can see, it is thicker than the flagella and thicker than the fimbriae, and can conjugate between two cells. This bacterial cell here, you can see the difference between here is the flagella, here is the pili, and sorry, it's the flagella who is the thickest, then the pili, then the fimbriae are many in number, but thinner and shorter than the two other appendages. I think we can stop here for this lecture, and next time we can start by the cell envelope and the difference between the two types of cell envelope in the gram-positive and the gram-negative bacteria. Thank you.